Every good experiment should be asking a question. If it's just kind of exploring some random thing for no particular reason, why are they spending all that money? Functional MRI is expensive, right? Okay, so in this little simple experiment, we're asking a question. The question is, are there any parts of the brain that are specialized for perceptually processing scenes? Okay, I mentioned this question before. Scenes more than other kinds of visual stimuli. Okay, that's the question. Okay, and if so, where are they in the brain? Okay, well, there are lots and lots of ways you could do a functional MRI experiment to ask this question. But here's a super simple one. Okay, so this is a diagram of an experiment that will run on me later tonight and has been run on many subjects before. This is what happens over time. You pop the subject in the scanner. They watch visual stimuli that are projected from outside the scanner onto a mirror right above the subject's head. The subject looks up at the mirror to see the stimuli, watches the stimuli while you scan their brain. Okay? And so what we want is to show the subject pictures of scenes like these. Okay, indoor, outdoor, some unfamiliar deserty scene, lots of different kinds of scenes. And we show them lots of different kinds of objects. Okay, this experiment here will be about, I don't know, I forget exactly how we put it together, 4 or 5 minutes long. And it will be composed of 18-second periods. First, where subjects are just staring at a dot, and then where they see a whole bunch of scenes flashing on the screen at a fairly rapid rate. The visual system can work really fast, so make it work, flash up lots of scenes. Okay? And then the subject looks at a whole bunch of objects flashing on a screen, and then a whole bunch of scenes and a whole bunch of objects. Okay? They can have different instructions. One instruction is just hold still, don't move, look at the stuff. That's one of the most common instructions we use in my lab. In Hillary's lab, they have to have more complicated instructions than that. Maybe not always. Sometimes you can just look at the stuff, right?
I work with kids, so we just make them listen to stuff.
Okay,
yeah.
Okay, that's good. That's good. Those, yeah, the hold still is the hardest part. Okay, so to figure out your scanning parameters, there's a bunch of choices you need to make. And you'll see all this happen. My lab tech, Janelle, will pop me in the scanner while Hillary narrates. And they'll first do a quick scout scan to get blurry pictures of my brain, just to kind of get organized. And then they're going to choose some slices to scan in. So for example, here's a subject, blurry picture of the subject's head. This is an angle through the brain like this. And here we've selected a bunch of vertical slices, also known as coronal slices, vertical slices like this, shown right here. Okay, so that's what, 8, 9 slices like that. Okay, so that's the first thing you do, is choose your slice orientation and thickness, and what part of the brain you're covering. In this experiment, we're looking only at the back of the head. This is a vision experiment. All this stuff back here is vision. Okay? In other experiments, you might want to look at the whole brain. There are various trade-offs. Sometimes you get lower resolution. If you want to look at the whole brain, there are many issues to consider. Okay? Okay, so we have the number of slices, their position, the thickness of the slices, and the orientation. These are all parameters you need to decide on to run an experiment. Okay. So this is just terminology for different slices: coronal, axial, sagittal. Okay? You'll see these things in papers, and you just need to know how they're scanning. Okay, so here is now one of those slices. This is an ancient slide, so this is a really blurry functional image from 15 years ago. They'll look better. So does this make sense? So this picture here is a slice through a brain like this. These lines are showing that the slices chosen are on this angle, right? And here is one of those rotated. Everybody oriented here? Okay, so this is the bottom of the brain. What's this down here? Cerebellum, yeah. And here's the brain up there. Okay, so that's a coronal slice. Okay, and so in a typical MRI experiment, you take that whole set of slices, a whole set of pictures, in 2 seconds. And then you do it again and again. Okay? That can vary. Sometimes you do it in 1 second or 3 seconds or something like that, but a second or two, something like that. Okay? To get the whole set. So what that means is, for each slice, like this one, we'll have a picture once every 2 seconds of that slice. And we'll also have a picture once every 2 seconds of all the other slices. So each slice is like a stack of images taken over time. It's a movie of that slice in the brain. Okay? And remember that the brightness of each pixel in each functional image is very indirectly related to neural activity at that region. Okay? Okay. So I'm just trying to explain the format of the data. So here's that slice in a stack over time. The time between successive images of the same slice is called the TR for repetition time. You'll see this in papers. And here is an example pixel or voxel. A voxel is just a three-dimensional pixel. Okay? So that pixel exists in each of these slices. And we could plot what that pixel is doing over the time of the scan. We could just take that pixel, pull it up in MATLAB, and plot it. And it would look something like this. Okay, is everybody with me? We've now taken one little voxel in the brain, and we've just plotted the magnitude of its MR signal, which is very indirectly related to neural activity, over time during a scan. This is just made-up data, but real data doesn't look much better than this. MRI data are very noisy. Okay, but everybody understand what we're doing here? We're just turning the data around different ways so you can understand what it looks like. Okay, so now remember we started with saying, are there any parts of the brain that respond more when you look at scenes than other kinds of visual stimuli? And the whole time subjects are, that we're collecting these data from that voxel, subjects are looking at different things. So we want to know whether that response is higher during the times the subject was looking at scenes than during the times they were looking at other things. So what we need to do is relate this time course to the paradigm that we put in there, right? So this is what the subject was seeing. This is now the same time course, and this is time. They were staring at a dot for 18 seconds. They were looking at scenes for 18 seconds, looking at objects for 18 seconds, and that's what that pixel was doing. Okay? So then what we need to do is statistically test whether the magnitude of that MRI response was higher when the subjects were looking at scenes than when they were looking at objects. Okay? So I'm going to leave all those details out for now. Hillary's going to lecture on that in a couple of weeks on how you actually do those statistics. You can think of it for now as basically a t-test or some other simple thing. It'll get fancier later, but it's just conceptually, it's a bunch of data points collected from the green epochs, and we're just saying, are those data points, are those magnitudes higher than these magnitudes? Okay? That's essentially all we're doing. Okay, everybody with me? Okay. So when you do that, here's what you see. This is a coronal or vertical slice in a subject, possibly me, I can't remember, from a very long time ago. And this shows activation in the brain of this subject, regions where this is a statistical result here. The color are the regions where the statistics said the MRI signal was higher when the subject was looking at scenes than when they were looking at objects. Okay? Okay, so for now, you can just believe those statistics. We'll get into the details later about how that's done and whether you should believe them. But for present purposes, let's just believe them and say, okay, in this experiment, those parts of the brain produced a stronger signal when this subject was looking at scenes and objects. Everybody get how this emerged from that experiment? Any questions about that? Okay, so now, yep.
Are you taking the point as your baseline? What is your baseline?
Okay, so this is a comparison of scenes versus objects. So actually in this analysis, that got dropped out.
So
why did
you do it?
Oh, good question. You do it for all kinds of reasons. One is to debug your experiment. Like, let's suppose you did this, and instead of this really lovely, exciting, this is as good as it gets in functional MRI, right? Nice, tight blobs with high significance. Okay, instead of this, you see the most common result, gray brains. No significant differences. At that point, you're just like, oh, what the hell? You know, you're just kind of swimming in vagueness and you don't know what to do. One of the things you don't know if the subject was asleep, if the scanner was broken, if your codes got screwed up, you don't know what the hell's going on, right? One thing that really helps you figure out what's going on is you can say, okay, let's take a real sledgehammer effect. Let's take all of our stimulus conditions, scenes and objects, and let's compare them to staring at a dot. And if we don't see a massive kind of forest fire of activity over the whole back of the brain and visual cortex, then the scanner's broken or the code's wrong or the subject was asleep, right? So it helps you debug is one thing. There are other benefits we'll get to later. But for present purposes, it's in part because I skipped over all this. I mean, actually you'd use it in the statistics as well. I was doing a simplified version of the statistics, the way we did it 15 years ago, because I didn't know any better. But now everyone does fancier things than that, where you actually use those data in your statistical analysis. But we'll get to that later.